Today we are going to learn about microscope. The study of the microscope is known as microscopy. So here the microscope is mainly invented by the scientist Leeuwenhoek in 17th century. So this microscope is an instrument which is used to see the tiny objects, organisms that are invisible to the naked eye. So here is the simple diagram of the microscope here. It mainly contains various parts like ocular lens, body tube, arm, objective lenses, stage, condenser, diaphragm, coarse and fine adjustment knobs, light source and a base. We will see one by one clearly here. The first one is the eyepiece. It mainly contains the ocular lens which mainly remagnifies the image formed by the objective lens. Here is a body tube which mainly transmits the image from the objective lens to the ocular lens here. So what is the objective lens means? It is a primary lens that magnifies the specimen. Here the magnification ranges from 10x to 40x. So here is a arm which mainly used to support the microscope when carried. It is nothing but a handle. So the next to the arm is the stage. So here this stage is to hold the microscope slide in a position, that is it will give supports to the slide being viewed. So below the stage there is another part, that is in a condenser, so which mainly focuses the light to the specimen. Below the condenser there is a diaphragm, so which mainly controls the amount of the light entering the condenser. There is an adjustment knobs like fine adjustment knob and coarse adjustment knob. This coarse adjustment knob is used to move the stage up and down for focusing, and this fine adjustment knob is used for to sharp the image. So there is a base in order to support the microscope. These are all the various parts of the microscope. And next one is the type of microscopes. There are various types of microscopes. The first one is the light microscope. This light microscope is again of so many types: bright field microscope, dark field microscope, phase contrast microscope, differential interference contrast microscope, fluorescence microscope. And the second type is confocal microscope, and third one is electron microscope. Again this is of two types, that is transmission microscope, scanning microscope, and the final one is scan probe microscope. So this is again of two types, that is scanning tunneling microscope and atomic force microscope. So these are all the various types of the microscope. We will see one by one clearly here. The first microscope is the bright field microscope. So what are the features of this is, it is mainly uses a visible light as a source of illumination. Here it cannot resolve the structure smaller than about 0.2 micrometers. Here the specimen appears against a bright background. It is very inexpensive and easy to use. Here the typical image which is used by using this bright field microscope is here like this. Coming to the principal uses, it is mainly used to observe the various stained specimens. It is also used to count the microbes. It does not resolve very small specimens like viruses. It is used to view up to microbes. So this is about the bright field microscope. And the next one is dark field microscope. The features are, it uses a special condenser with an opaque disc that blocks the light from entering the objective lens directly. Here the light reflected by the specimen enters the objective lens and the specimen appears light against a black background. So observe the typical image viewed by using this type of microscope is here like this. Principal uses, It is mainly used to examine the living microorganisms that are invisible in bright field microscope. At that time when the bright field microscope is not that much effective to view, so use this type of dark field microscope. So do not stain easily or destroyed by the staining. So at that time we will use this type of dark field microscope. At the same time, it is frequently used to detect the trypanosome palladium in the diagnosis of the syphilis. So all these are all the uses of the dark field microscope. So phase contrast microscope. Features are uses a special condenser containing an annual ring-shaped diaphragm. So the diaphragm allows the direct light to passes through the condenser, focusing light on the specimen, and a diffraction plate in the objective lenses. Here the direct and the reflected or diffracting light rays are brought together to produce the image here. There is no staining required for the specimen here. The principal uses are, it is used to facilitate the detailed examination of the internal structures of the living specimens. Living specimen can be easily viewed by using this type of phase contrast microscope. There is no staining required for this. So due to the contrasting phases phenomena, so it will view the image in a colored way. So this is the typical image which is viewed by using this type of phase contrast microscope. So this is about the phase contrast microscope, and the next one is differential interference contrast microscope, that is DIC. It is like in a phase contrast microscope. It uses a differences in the refractive index to produce the image. It uses two beams of light separated by a prism. The specimen appears colored as a result of a prism effect. There is no staining required. So principal uses are, it is used to provide the three-dimensional image, that is 3D view of the image. So the typical image is here like this. So by viewing with the differential interference contrast microscope, that is DIC, so the image will appear like this, that is in a 3D manner. And the next one is fluorescence microscope. Here the features are, it uses an ultraviolet or nearly ultraviolet sources of illumination that causes the fluorescent microbes in a specimen to emit the light. So here the typical image viewed by using this fluorescence microscope is here like this. The microbe will appear in green color due to the fluorescence effect. Here the principal uses of this type of microscopes are, it is for the fluorescent antibody technique, that is immunofluorescence, to rapidly detect and identify the microbes in a tissues or a clinical specimens. And the next one is confocal microscope. The features are, it uses a laser light to illuminate one plane of a specimen at a time. It is also like in a fluorescent microscopy, and the specimens are stained with a fluorochromes, so they will emit or retain the light. Here the typical image appeared by using this confocal microscope is like here. The principal uses are, it is used to obtain 2 and 3 dimensional images of a cells for biomedical applications. It is used to evaluate the cellular physiology by monitoring the concentration of the substances such as ATP, calcium ions, like that. And the next one is electron microscope. The features of the electron microscope are, it uses a beam of electrons instead of light. Here the electrons passes through the specimen because of the shorter wavelength of the electrons. Here the structure smaller than 0.0 micrometers can be resolved by using this type of microscope. So the image produced is a two-dimensional one. Here the typical image viewed by using this electron microscope is here like this. Uses, it is used to examine the viruses or the internal ultrastructures in a thin section of a cell. Usually magnifies 10,000 to 1 lakh x lengths. So this is about the electron microscope. And the next one is scanning microscope. The features of the scanning microscope are here like this. It uses a beam of electrons instead of light. Electrons are reflected from the specimen because of the shorter wavelength of the electrons. Here the structures smaller than 0.0 micrometers can be resolved by using this type of microscopes. The image produced appears three-dimensionally, that is in a 3D view. So here the typical image viewed by using this scanning microscope is here like this. Uses. It is used to study the surface features of the cells and the viruses, usually magnified 1000 to 10,000x. It can resolve the object as closer together as 20 nanometers. So it is a highly efficient microscope. And the next one is scanning probe microscope. It is again of two types, that is scanning tunneling microscope and atomic force microscope. So we will see the scanning tunneling microscope here. The features are, it uses a thin metal probe that scans a specimen and produces an image revealing the bumps and depressions of the atoms on the surface of the specimen. The resolving power is much greater than that of an electron microscope. There is no use of the special preparations in order to use this type of microscope. And the typical image viewed by using this microscope is here like this. Uses, it provides very detailed views of the molecules inside cells, resolve features that are only about 1 by 100 the size of an atom. It is more powerful and effective microscope. And the next one is atomic force microscope. The features are, it uses a metal and a diamond probe gently forced down along the surface of the specimen. Here it produces a three-dimensional image. There is no need of use of any special preparations. Here the typical image viewed by using this atomic force microscope is here like this. And uses, it provides image of a biological molecules in nearly atomic detail and a molecular processes such as assembling of the fibrin, components of the blood clot can be able to viewed by using this type of microscope. It is also a more efficient microscope. So this is all about the various types of microscopes. Thank you for watching this video. Please subscribe to our channel for more videos.